Hi guys, welcome back to Scientifically Speaking, and today we are gonna dive into one of the most common examinations we do not just in cardiology but just like in general. We do this like a lot, and it's called ECG, aka electrocardiography. So let's take a look. What's important to know here is that our heart has some specialized cells, cardiomyocytes, that are able to generate action potential by themselves. The cells are like, they look something like this. It's marked with yellow here in this picture, and thanks to that, this right here, this structure is generating the potential, leading it further to the heart, and that leads to contraction of the heart, because we know that we need that innervation for heart to pump like 24/7, and we need it to pump blood for survival, obviously. So that's just like to sum it up. And also we know that our heart is influenced by parasympathetic and sympathetic nervous system, but they just like alternates the function of its own nervous system. So that's just like we know the basics and we can move on. Right, so this is the typical ECG wave or waves, or how we just like can how we see it like on people's tattoos and t-shirts and whatever. But this is how it looks, and today we are gonna explain like why it looks this way, why this is like the physiological rhythm, and how the ECG should look graphically. So now, what's important to mention as well is that what ECG is, it's pretty much potential on the cell membrane. So you can see a cell here. This is a cell, and we know that we've got some like intracellular and extracellular ions. While the main ion inside the cell is the kalium one, and outside of cell it's natrium and chloride. So these are the just some basics of like our cells, and just the main ions of the cells inside and outside, and they create something called membrane potential. It's like right here on the cell membrane. And when this potential changes, for example, if one of these cells that's marked yellow here generates the action potential, it changes, right? It goes from minus to plus, and then it spreads, and thanks to that spreading, we can contract the myocardial cells. So that's one thing, just like, so you know, you are aware of that, you know these basics. Now we've got like the frames here. It represents the cardiomyocytes, the cells of the heart, of that like action potential that the specialized cells generate in our heart. And we also place some of the electrodes on the human body, on the surface, right? Because we are measuring what's outside, because we cannot like measure, or we can measure what's inside, but we would have to like, it would be more invasive method, because you would actually have to put the electrode inside of the cell, and it's like not as easy as just like measuring it outside, measuring the potential outside. So that's why we've got these electrodes here, and we are measuring it outside of the cells. So that's important to remember. Just note that we are measuring outside of the cell always when it comes to ECG. So we've got like some basic state here. First, everything is negative, nothing is happening, no action potential is generated. So this is like, what am I talking, like inside the cells. Meanwhile, outside, everything is positive. We've got lots of pluses, and it's just like the basic state. So that's how it is. Because here, the inside is negative, because we've got lots of like negative charged proteins inside of the cell, while the outside is positive, because the concentration of natrium outside of the cell is like massive. It's like really, really a lot. Right. So this is like the beginning. This is where we start. We are measuring the outside of the cells, in relation to these electrodes that we have right here. But like now, when everything is the same, nothing is happening, we've got something called isoelectric line. It's just flat. It's just flat. Nothing is happening. You can see that in the movies, that like when nothing is happening, the person is dying on the operating table, it's flat. The line, the ECG line is flat. But what happens when the impulse goes a bit further? It goes from here and it starts something in the cells. Then something like an event called depolarization occurs. How does it look? Well, it starts being positive inside, but just like part of it. The rest is still negative. It's still remaining negative. Well, outside, it's negative because like it switches, like thanks to the ions going inside and outside of the cell, cations and anions, and them going in and out. It just alternates the potential, the membrane potential, and the inside of the cell becomes more positive, as you can see right here. And the rest is still negative, but we've got positive charge outside. You see like the half of it remains the same and the first half is changing, thanks to the nerve impulse. Now, what can we see here? It's important to note that I'm gonna just like draw some arrows here. And the arrows, they always go like from the positive charge inside to the negative charge inside. So we go from positive, it's like the vector, positive to negative. So this is our arrow when depolarization occurs. I'm gonna write it right here, depolarization. Now, what can we see here? What matters are also the electrodes that are outside. If the arrow is from negative electrode to positive, then we can see a wave that is going up. So something like that. Or wait, I'm gonna just draw it prettier. Something like that. If it's the other way around, from positive to negative, it's gonna be down, but we will dive into this a little later in this video. Now, what happens next? Next time, everything inside, because the depolarization spreads, so everything inside is going to be depolarized, aka positive. Meanwhile, everything outside is going to be negative, as I'm just drawing right here. How does this look like on the ECG line? Well, since everything is same, there's like no change. Everything's just like same inside and outside. It's gonna be isoelectric again, so flat. It's gonna be flat. What happens next? You guessed it. The beginning of the cells here is starting to go. Is starting to be repolarized, which means it's turning back to the original state which you have here in step 1. So it's going to be negative again, while the rest is still going to be positive because it didn't have time to go negative yet. And yes, we can just continue like this. How does the vector look like here? Well, as you can see, we always go from I'm talking about what's inside now, plus to minus. And we are going to focus on the electrodes here, the plus and minus, and we can see that it goes from plus to minus, which means that it's gonna be negative under the main line, the wave. Okay? Now we are gonna I'm gonna show it to you on the heart so you can imagine it better. But this is the basics that you need to understand before we go further. So you can see the specialized cells in the heart here. Again, you can see the bundle of His, the Tawara parts and the Purkinese parts of the system. And so, as I said, we are measuring what's outside. So when depolarization occurs, it's going to be negative outside, right? Because it's positive inside, so negative outside. That's what's important to realize. So when we have the potential generated here, it's going to be positive inside the cells, but we are measuring what's outside, so we are gonna measure the negative. I'm always gonna draw here what's outside. We also have the electrodes here again. So now we still have this part not depolarized yet, so we can measure plus outside of the cells. As I said, outside, it's important to know that. And we are going from minus to plus. Therefore, the P wave, aka the depolarization of atria, is going to look like this, and it's going from minus to plus, so the wave is going to go up, the ECG wave. We are gonna just draw it here. So this is the P wave. This is the first one. What's gonna happen next? We need to depolarize the ventricles. It's like the biggest mass of the heart, the ventricles. So we are gonna do it like slowly from the septum. The septum is right here. What is happening here is that the left part of the septum is depolarized first. So we've got minuses here first, and then the right part is depolarized a little bit later. Right? So the arrow here for the Q wave is not gonna be like exactly straight up or down, because like that's not just how it works, but it's gonna be a little bit moved towards the right part. Like from our point of view, it's the left. If the left is the left side, but it's gonna be again from minus to plus. So imagine it's something like this, and we can see that like on the main heart axis, which is like this, it's going to be from minus, or from plus to minus side. So that's why the Q wave, it's going to be a little bit like this, and it's going to be going down. So that's the Q wave. Now we need to depolarize more of the ventricles. So how is that going to look like? Well, we are going to start depolarizing this part of the heart. You know what? I'm just gonna draw it here in the separate picture so we can see it better, because it's going to be a little bit more complex. So the rest is still not depolarized. And we are gonna have like two vectors here, because it's going again from minus to plus, and now we are gonna just like connect the vectors, and we are gonna get the main vector, which is the R wave. So we've got PQ, RS. QRS complex means depolarization of the ventricles, but what we have to keep in mind is that still like the repolarization of atria is happening here. It's just like not visible in the ECG waves, because it's like smaller compared to depolarization of ventricles, because they have more muscle mass, therefore it's like bigger. So this is the QRS complex again, which means that our ventricles are depolarizing. That's the R wave. You can also see that we go from minus to plus on the R, therefore we go up for the R. Now the S wave, I already drew it, but now we are gonna take a look at like why is that happening. So why is that happening? Because here the depolarization starts from here. Everything this is like going on also because we need to eject as much blood as possible, and we need it to be effective. So the cardiomyocytes, the contractile one, they contract from the bottom of the heart for like efficient blood ejection. Note that also, it's also important kind of. So we are working on the S wave now, which is gonna look like this. And as you can see, it goes from plus to minus, the arrow, because here, like inside the heart, what I marked is that it goes from the minus to plus again, but on the main axis you can see it goes from plus to minus. That's why the S wave is going to be going down. What happens next? We've got like one more wave remaining, and that is T. We are not exactly sure what the T wave is doing, but it's here, and I'm gonna just like paint it here or draw it here, because the T wave, it's pretty much this again. So the T wave goes like that. Right, now we've got the full sinus rhythm on the ECG draw here. So that's how it looks. I think that's it. So I hope you understood. I hope that helped. It's like important cardiac physiology, and I know that like sometimes it's easy to get a little lost in that, but this is like the best explanation that I can provide. This is just like how I understood it in medical school, and it's really important, so you better learn it, and hope it helped. Don't hesitate to subscribe, like, and comment, and I'll be happy to see you here next time.